Good morning, students. Today we are going to learn a new chapter, chapter number 4, Principles of Inheritance and Variation. Inheritance and variation we learn under genetics. So, genetics is a branch of biology that deals with the study of heredity and variation. What do you mean by heredity? So we get the characteristic features from our parents. When a baby is born, we used to compare the features of the baby with their parents. Like the nose is like her mother, or the mouth is like her father. Like that, we used to compare because we get the characteristic features from them because they contribute these characters in the form of the chromosomes. And so variation also, we differ from the parents in certain characteristic features to certain degree, and this we call it as variation. And so genetics describes how the characteristic features pass on from the parents to their offsprings in each successive generation. Now, what is a gene? Gene is the unit of heredity. The unit of heredity is known as gene. Where is this gene present? These are present in the chromosomes, and chromosomes are present in the nucleus. A gene is the inherited factor that will determine the biological character of an organism. So this is the definition of gene. Gene is the inherited factor that determines the biological character of an organism. What is variation? A variation is the degree by which the progeny or the offspring differ from their parents. To how much they differ from their parents is the degree by which the progeny or the offsprings differ from their parents. Now let us pass on to multiple alleles. So before learning what are multiple alleles, we have to recall our previous knowledge. We have learned about the Mendelian genetics. In that we have learned about the tallness and shortness. So tallness or shortness, they are determined by two alternative forms of the same gene. So every gene has two alternative forms, and they will segregate and stand at the time of gamete formation. So the tallness or the dominant character is represented by capital T, and the dwarf or the recessive character is represented by small t. So the former tall is a normal allele or wild allele we can say, whereas the small t is mutant allele. A gene can mutate several times producing several alternative forms. Here we are seeing only two forms, two alternative forms. When the same gene is going to have, if it mutate to have several times to produce several alternative forms, we call that as multiple allelism. So what is meant by multiple alleles? A gene that can mutate several times to produce several alternative forms of the same gene is called as multiple allelism. So, when three or more alleles of a gene controls a particular trait that occupies the same locus on the homologous chromosome, I hope you all know what are homologous chromosome of an organism. They are called as multiple alleles. So, and their inheritance is said to be multiple allelism. So, homologous chromosomes, we get this pair, one from father and another one from mother, but they will determine the same type of characteristic features for the progeny. So, when three or more alleles of a gene control a particular trait that occupies the same locus on the homologous chromosome of an organism, they are called as multiple alleles, and their inheritance is said to be multiple allelism. So multiple allelism, the best example for it is human blood groups. So particularly in the inheritance of different types of blood groups, multiple allelism we can study. So blood group inheritance in human can be understood by learning about the antigens and antibodies. We all well know antigen A, antibody B, antigen B, antibody A will be present in the particular blood groups. In the multiple allele inheritance of ABO blood groups, blood differs chemically from person to person because of the presence or the absence of the antigen. When two different incompatible blood types are mixed, agglutination takes place. What is meant by agglutination? Agglutination is clumping together of the erythrocytes. So the basis of these chemical differences is due to the presence of the antigen on the surface of the RBC and the epithelial cells. Carl Landsteiner discovered the two kinds of antigen called antigen A and antigen B on the surface of the RBC. Based on the presence and absence of these antigen, three kinds of blood groups are there. Type A group, B group and O group. When antigen A is present, A group. When antigen B is present, B group. When no antigen is present, it is called as O group or universal donor were recognized. So after in 1902, two students of Landsteiner found that AB group, when a person has both the antigen A and B, that group is called as AB group, and this was discovered by D. Canstelle and Sturley in 1902. And these are the precursor cells of the RBC. Before the RBC matures, it undergoes these different stages: pre-erythroblast, basophilic erythroblast, polychromatic, orthochromatic and polychromatic erythroblast. Bernstein in 1925 discovered that the inheritance of the different blood groups in human beings is determined by a number of multiple allelic series, and these multiple alleles were found in the chromosome number 9, and they are the autosomal alleles. Autosomal alleles means they determine the body characters. And the gene controlling blood type has been labeled as L after the name of the discoverer Landsteiner, and I from the isoagglutination. And so the gene I exists in three allelic form, that is IA when antigen A is present, IB when antigen B is present, IO no antigen is present. IA specifies antigen A, IB antigen B, and IO specifies no antigen. The individuals who possess these antigens in their fluids such as saliva are called as secretors. So when the person is having these antigens in their body fluids like saliva, they are said to be called as secretors. It's an important question. Formation of A antigen. So IA allele produces an enzyme which is called N-acetyl galactose transferase, and it can add N-acetyl galactosamine. So we call in short as NAG. Same way, the person is having B antigen, IB allele encodes for the enzyme galactose transferase, and it can adds the galactose to the precursor to form the H substance. So formation of H antigen, each allele IA and IB produces transferase enzyme. In case of IO, that is no antigen, no terminal transferase enzyme is produced, and therefore it is said to be null allele, and hence cannot add any NAG or galactose to the precursor. So from the phenotypic combination, it is evident that the allele IA and IB are dominant to IO, but both of them are co-dominant to each other. So their dominance hierarchy can be given as IA equal to IB, which is greater than IO. A child receives one of these alleles from each parent, giving rise to 6 possible genotypes and the 4 possible blood types, which is called as phenotype, and 6 genotypes and 4 phenotypes. The genotypes are IA-IA, IA-IO, IB-IB, IB-IO, IA-IB and IO-IO. Are you able to understand this? So here one of these allele will be one from father and one from mother we will get. So father may give A and mother may give O, that is also to the blood group A. So father and mother both may contribute IA allele, and so also A blood group. In the B blood group, father may give B gene and allele, and mother may give O null allele, and so the blood group is B. Same way, if both are contributing B allele, also the child's blood group will be B. If father is giving A and mother is giving B, then the child's blood group will be AB. So it can also be vice versa. So we can get B from father and A from mother also. So then the child's blood group will be AB. If both are not giving any antigen, then IO it becomes O blood group. So both the alleles are expressed in the phenotypes. A and B are codominants. Now let us learn the rhesus or the RH factor. RH factor or RH antigen is also found on the surface of the erythrocyte along with antigen A and antigen B. And this was first discovered by Karl Landsteiner and Alexander Wiener in 1940 in the blood of rhesus monkey Macaca rhesus. And later it is found in the human blood also. The term RH factor is referring to immunogenic D antigen. If the individual is having this D antigen, then the person is said to be RH positive. If the person does not possess this D antigen, then the blood group is RH negative. Rhesus factor in the blood is inherited as a dominant trait. So naturally occurring anti-D antibodies are absent in the plasma. So antigen may be present on the surface of the erythrocyte, but antibodies are absent in the plasma of any normal individual. But however, when the person RH negative person is exposed to RH positive blood group, then he or she will tend to produce anti-D antibodies. On the other hand, when RH positive person receives RH negative blood, then there won't be any effect. The genetic control of RH factor. Fisher and Ray's hypothesis. RH factor involves three different pairs of alleles located on three different closely linked loci on the chromosome pair. So it involves three different pairs of alleles located on three different closely linked loci in a chromosome pair. And this system is more commonly in use today. And it uses CDE nomenclature. Let us see what is CDE. So here you can see the three pairs of RH alleles. Capital C, small c, capital D, small d, capital E and small e in the same chromosome pair. So they can link or join in any way. So the possible genotype will be capital C or small c, or one capital D or small d, or one capital E or small e from each chromosome will join, and so the configuration may be like this: capital CDE, small cde, or capital C, small de, capital E, small c, capital D, small e, and so on. So all these genotypes carrying D, capital D allele will produce RH positive. And so here you can say this genotype and this genotype and this genotype will be RH positive when there is double small d, that is recessive genotype, double recessive genotype DD will give rise to the RH negative phenotype. So here for your understanding, better understanding, it is been enlarged how the chromosomes, the alleles on the chromosomes can join. Here now let us see the Wiener's hypothesis. So here Wiener's hypothesis, haplotypes. Haplotypes means single gamete chromosomes. When Wiener proposed the existence of 8 alleles, the above said we had only 3 pairs of alleles. Here it is 8 alleles. That is R1, capital R1, capital R2, capital R0, capital RZ, and small r, small r1, small r11, small ry. at a single locus, single RH locus, say any one of these allele will be present. So all these genotype carrying the dominant R will be RH positive, and all the genotype which is having double R, recessive double R, will be RH negative. Now incompatibility of RH factor. So erythroblastosis fetalis, RH incompatibility, has great significance in childbirth. If a woman is RH negative and if he marries a man with RH positive, the fetus may be RH positive because it would have inherited from its father. So RH negative mother becomes sensitized by carrying the RH positive fetus within her body. Due to the damage of the blood vessel during the childbirth, the mother's immune system recognizes the RH antigen from the fetus blood and gets sensitized. The sensitized mother produces RH antibodies, that is anti-D antibodies will be produced by the mother blood. The antibodies are IgG type, so which are very small and can cross the placenta and they can enter to the fetal circulation also. So by the time the mother gets sensitized and produce anti-D antibodies, the child is delivered. So for the first delivery, the mother and the child will not have any problem. Usually no effect is associated with the exposure of the mother to the RH positive antigen for the first childbirth. But subsequent RH positive children carried by the same mother may be exposed to the antibodies produced by the mother cell against the RH antigen. And so there will be problem, and agglutination will take place from the second child onwards. This causes the hemolysis of the fetal RBC and resulting in the hemolytic jaundice or anemia, and this condition is called as erythroblastosis fetalis or hemolytic disease of the newborn, HTN in short. To prevent this erythroblastosis fetalis, if the mother is RH negative and the fetus is RH positive, anti-D antibody should be administered to the mother at 28th and 34th week of gestation as a prophylactic measure. Prophylactic means munnecharikai. So if the RH negative mother delivers RH positive child, then anti-D antibody should be administered to the mother soon after the delivery. So this develops a passive immunity and prevents the formation of the anti-D antibodies in the mother's blood by destroying the RH fetal RBCs before the mother's immune system is sensitized. So this has to be done whenever the woman attains pregnancy. Now let's see what is sex determination. Sex determination is the method by which a distinction or the differences between the male and the female is established in a species. It is initially established by the chromosomes, and these chromosomes are called sex chromosome. And sex chromosome determine the sex of the individual in the dioecious or unisexual organism. Dioecious means male separate body organism and female separate organism, and this we call as unisexual organisms. Best example of it is human being. Chromosomes other than the sex chromosomes of an individual are called autosomes. So there are totally 23 pairs of chromosome in human beings. Out of 23, 22 are called as autosomes. They will determine the other body characteristic features, whereas the last pair, 23rd pair, is called as sex chromosome. The sex chromosome may be similar or dissimilar. If it is similar, it is called as homomorphic. So in female it is similar and it is called as homomorphic, and it is determined by XX double X. So it is homomorphic. When it is dissimilar, it is called as heteromorphic. In male it is XY, and so it is heteromorphic. The individuals having homomorphic sex chromosome produce only one type of gametes, that is A plus X, AX. Whereas the heteromorphic individuals will produce two types of gametes, that is AX and AY. So here you can see the homomorphic individuals and heteromorphic individuals. So chromosomal basis of sex determination. Heterogametic sex determination. In heterogametic sex determination, one of the sex produces similar gametes whereas the other sex produces dissimilar gametes. The sex of the offspring is determined only at the time of fertilization. So heterogametic males. So in this method of sex determination, the males are heterogametic and produce dissimilar gametes, while the females will be homogametic, produce similar gametes. So it is of again two kinds. It can be XXXO type or XXXY type. Types of sex chromosomal mechanism of sex determination. Heterogametic males. So heterogametic males will be of these two types, whereas heterogametic females will be of Z2 ZZ system or ZW ZZ. Here the females will have the heterogametes Z and O, whereas the males will have the similar gamete, that is Z. Now let's see the XXXO type. This method of sex determination is seen in bugs, some insects like cockroach and grasshoppers. The female will have two X chromosomes, whereas the male will have only one X chromosome, and it is heterogametic. The presence of the unpaired X chromosome determines the male sex. The male with the unpaired X chromosome produces two types of sperms, one half with X chromosome, another half without X chromosome. The sex of the offspring depend on the sperm that fertilizes the egg. If the A plus O type fertilizes the egg AX, then it will be male. When A plus X sperm fertilizes the AX egg, then it will be female. So this is the schematic representation of XXXO type enlarged for you. Next heterogametic male type is XXXY type or ligaeus type, and this method of sex determination is seen in human beings and Drosophila. The females are homogametic with the XX chromosome, while the males are heterogametic with the XY chromosome. Homogametic females can produce only one kind of egg with each X chromosome in it, whereas the males can produce two types of sperms with the X and Y chromosome in it. So while the heterogametic male produces two kinds of sperms, the sex of the embryo depends on the fertilizing sperm only. If an egg is fertilized by X bearing sperm, then the individual produced will be female. If the egg is fertilized by Y bearing sperm, then the males are produced. Next we are going to see the heterogametic females. Here in this method of sex determination, homogametic males will possess two X chromosome, whereas the females will produce the dissimilar gametes. Female sex consists of single X chromosome or one X and one Y chromosome. So this type of sex determination is seen in certain insects, fishes, reptiles, birds, etc. So thus the females are heterogametic and produce two types of eggs, whereas the sperms produced will be of single type. To avoid this confusion, the XXXO and XXXY types are changed with the alphabet as Z for X and W for Y. So heterogametic females are of two types, ZO ZZ type or ZW ZZ type. So this is the heterogametic female system of sex determination. So ZO ZZ type. This method of sex determination seen in mouse, butterfly and domestic chickens. So in this type, the female possesses Z chromosome in its body cells, and heterogametic ZO produces two kinds of eggs, Z chromosome or without Z chromosome, and the males will possess only Z chromosome and it is homogametic. So when they fertilize AZ female eggs, then the individuals produced will be male, and AZ sperm, when it is fusing with the A0 eggs, then the individuals produced will be female. ZW ZZ type. This method of sex determination is seen in insects like gypsy moth, vertebrates, fishes, reptiles and birds. In this method, the female has only one ZR and one W chromosome producing two types of eggs, whereas the male will produce single type of sperm, that is Z type. So when the sperm which is having Z, when it fertilize the egg Z, then the individual produced will be male. When the sperm fertilize the W is egg, then the individuals produced will be female. Sex determination in human beings, which we have already learned, this is just a recalling. So genes determining the sex in human beings are located in two sex chromosomes which are called as allosomes. In mammals, sex chromosomes are associated with the chromosomal differences between the two sexes. So typically XX for female and XY males. 23 pairs of human chromosomes include 22 pairs of autosomes, that is 44A, A we say, and one pair of sex chromosome is XX or XY. The females are homogametic, producing only one type of gamete X, or each containing X chromosome, while the males are heterogametic, producing two types of sperms, X or Y chromosome. An independently evolved XXXY system of sex determination is seen in the human beings as well as in Drosophila. Now let us learn about the Y chromosome and the male development. Current analysis of the Y chromosomes have revealed numerous genes and the regions with the potential genetic function. So some genes with or without homologous counterparts in the X chromosome. So present on both the ends of the Y chromosome are pseudo-autosomal region or pars. Pseudo-autosomal region, this yellow color is pars, or the pseudo-autosomal region on the Y chromosome. And these are similar and they are present even in the X chromosome. And this can get synapse and they can recombine during meiosis. The other 95% of the Y chromosome is referred as non-combining region or NRY. So they will not be combined with the Y chromosome during meiosis. And so it is called as non-combining region of the Y or NRY. The NRY is divided equally into functional genes and non-functional genes. Functional genes are called as euchromatin and non-functional genes are called as heterochromatin. So within the euchromatin region there is a gene called sex determining region. In human, if the Y chromosome is absent, it leads to the development of the female because the SRY gene is absent in the X chromosome. Because this SRY is responsible, the gene product of SRY is responsible for the testis development, or testis determining factor is present in the SRY, and that is present in the adult male testis. What do you notice about the difference between the X and the Y chromosome? The Y chromosome is much smaller whereas the X chromosome is little longer. The Y chromosome carries the SRY gene. This gene is called a sex determining gene in the euchromatin region of the Y chromosome because it causes the male sex organs to develop. Now let us see about the dosage compensation or Barr body. Barr body in 1949, Barr and Bertram, they both first observed a condensed body in the nerve cells of the female cat, which was absent in the male. This condensed body they called it as sex chromatin, and later it was referred as Barr body. In the XY chromosomal system of sex determination, males have only one X chromosome, whereas the females will have two. So a question rises, how does the organism compensate for this dosage differences between the sexes? In mammals, the necessary dosage compensation is accomplished by the inactivation of the X chromosome in the female, so that both male and female have only one functional X chromosome per cell. So here you can see Mary Lyon suggested that Barr bodies are represented as inactive chromosome, which is in female it becomes tightly coiled into heterochromatin, a condensed and visible form in the chromatin. The number of Barr bodies present in the cell will be 1 less than the number of the X chromosome. That means if there are 2 X chromosome, the number of Barr bodies in the cell will be 1. If there are 3 X chromosome, the number of Barr bodies will be 2. So this we can understand betterly by this. The XO female have no Barr body, whereas the XXY males will have one Barr body. Here when you see XX female, it is normal in human beings. XX female will have only one Barr body because one X become active, and another X is represented by the bar body. Whereas XY male will not have any bar body because only one active X is present here. Whereas XXY male Klinefelter syndrome, one X become active whereas the other X become the bar body with the Y chromosome. And XXXX female will have two bar bodies because only one X become active X here. Next we have the haplodiploidy in honeybees. In hymenopteran insects such as honeybees, ants, wasps, a mechanism of sex determination called haplodiploidy mechanism of sex determination is common. In this system, the sex of the offspring is determined by the number of sets of the chromosome it receives. Number of sets, how many sets of chromosome it receives, that determines the sex of the individual. The fertilized egg develops into female, queen and worker. So from the fertilized egg develops a female, either queen or workers. Whereas the unfertilized egg will develop into males, which is called as drones. And this process we call as parthenogenesis. Without fertilization, egg develops into male drones. It means that males have only a half the number of chromosome, that is haploid, whereas females will have the double number of chromosome diploid. Hence the name haplodiploidy is given for this system of sex determination. This mode of sex determination facilitates the evolution of sociality in which only one diploid female becomes the queen and lays the eggs for the colony. All the other females which are diploid, having developed from the fertilized eggs, will help to raise the queen's egg and so contribute the queen's reproductive success and indirectly to their own success, a phenomenon known as kin selection. So the queen constructs their social environment by releasing the hormone that suppresses the fertility of the worker. So even though both workers and queen are developing from the fertilized egg, that is diploid cell, the queen will release a hormone that will suppress the fertility of the workers, and so it will be maintained as the female. So here you can see egg, when it is fertilized, it can develop into worker or queen, whereas the unfertilized egg will develop into drone.